Hi everyone, my name is Vala and I'm an alumni of Boston University Biomedical Engineering Master's Program. BU BME department was founded in 1966. It is one of the largest BME departments in the US with around 100 primary and affiliated faculty in diverse research domains like biomechanics, biomaterials, imaging, computational modeling, molecular and cellular and tissue engineering, neuroengineering, system and synthetic engineering, nanotechnology, and sensing. Both of its undergrad and grad programs are consistently ranked among the top BME departments in the nation by US News and World Report. I am collaborating with the BME GSC student organization to interview distinguished faculty in the BU BME program. We want to learn about their research, academic journey, and their insights and advice for BME undergrad, grad students, and international students. We want to understand what makes them distinguished and how they think. These conversations are incredibly valuable, so we'd like to share them with you. If you enjoyed this video, please give us a thumbs up and feel free to share. We want as many people as possible to benefit from this content. If you don't like it, give us a thumbs down because we'd like to know that as well. Today, our guest is Professor John White. John is professor and chair of BU BME department. He employs engineering approaches to better understand how information is processed in the brain. Combining computational modeling, electrophysiological and optical techniques, and imaging methods, he and his lab have worked to advance new neuroscience technology to understand memory disorders and epilepsy. John received his Bachelor of Science degree in BME from Louisiana Tech University and his PhD in BME from Johns Hopkins University. As a principal or co-principal investigator, John has raised over $50 million in grant funding from the National Institute of Health, National Science Foundation, private foundations, and other sources. He is the principal investigator and program director for BU BME's long-standing NIH training grant in quantitative biology and physiology. He is the president of the Biomedical Engineering Society. John was also the co-director of methods in computational neuroscience in Marine Biological Lab. He is an elected fellow of American Institute for Medical and Biological Engineering. One of my friends works in John's lab, and he told me that John is a very caring person. He cares about his students, and he always focuses on how to better serve and improve the department, how to create an impact and make a difference in the lives of BME students and faculty. That's why I feel truly blessed to have him as the department chair. John, thank you so much for your time today.
Yeah, my pleasure.
So in our first part, we're going to talk about your research and the projects you're working on right now. So could you give us a brief overview of your lab and then what projects are you working on and then why are they significant?
Yes, I'm happy to. So I'm trained as mainly an electrical engineer, and we take those approaches into the study of neurophysiology, neuroscience, the functioning of neuroscience, and we're working toward clinical gain in those kinds of problems. So we use a lot of engineering approaches, including imaging techniques, computational modeling, electrophysiology, etc. And so we're working on a number of major problems, but I'll focus on a few. One is the biophysical basis, the mechanistic basis of how organized activity, which is so important for how the brain functions, is generated from the molecules up. So we've been working on this for several years, and we've made a number of useful contributions in that area. We're very interested in learning and memory. That's probably our main function, our main point of interest. And we also are interested in the basic idea of how we represent the sensory world and how we execute commands and drive muscles to achieve our goals.
That's very interesting. Could you tell us a little bit more about the memory project?
Yes, I'll give you an example of one of the memory projects. So we study a form of memory called episodic memory, which is autobiographical memory. So if you're telling a story later tonight about this moment, then that would be an episodic memory.
Gotcha.
And this episodic memory is stored in a part of the brain called the hippocampus, and it was actually made famous in a movie that from my day called Memento, which is actually a terrific movie a terrific movie that you should look up. It's probably 20 years old now. But it's laying down memories that we can then discuss and talk about, autobiographical memory. Now, some of this work is done in human subjects. We work in rodents. So they have episodic memories. They're mammals. They have episodic memories very, very much like us. So we're trying to understand the details of the encoding and decoding of memory. How is a memory laid down, and how might we design a prosthetic device in the long term that would help people with memory disorders improve their memory.
Oh, that's fascinating. Very interesting. What is the trend or most popular research domain in the neuroscience? And then what is the urgent question or the bottlenecks we are trying to solve, in your opinion, and what the community thinks in general?
Yes, those are great questions, and there are a lot of bottlenecks. I think that we're struggling to understand. In order to fix something, you have to understand it.
Exactly.
And we're trying to understand something that we're a bit out of our depth, honestly, in trying to understand the mechanisms of memory, the mechanisms of consciousness. And meanwhile, it's extremely devastating to families and lives and to the economy, neurological disorders. So Alzheimer's disease, for example, the population is growing older and older. My generation, the baby boomers, we're all starting to approach retirement age. The risk of Alzheimer's skyrockets in the 80s. Over age 80s. We are trying to understand these things. We don't understand the mechanisms by which cognitive problems like Alzheimer's or schizophrenia or you name it, depression arise. And but we have a lot of hints. Now, once we have a better basic understanding, we still have the problem of how do we interact with the nervous system to improve conditions. So we have some exciting things that have happened in the last 10 years. For example, this is not my work, but it's work in the field in general. There's some really exciting work about prostheses that enable people who are paralyzed to interact with the world by reading brain signals, usually via electrodes that are implanted into the brain or EEG or something like that, and then driving robotic devices to give people people who are paralyzed or have locked-in syndrome or ALS, a way to interact with the world. But we're not very good at the scientific and engineering communities. We're not very good at interacting with biology in a way that biology is comfortable with. And what I mean by that is you put electrodes in the brain and the brain generates this large inflammatory reaction. So we have a huge amount of work to do in terms of generating technology that's much more bio-friendly.
Right. Okay, I'm just curious. I think a couple months back, Elon Musk, he has a new idea about the Neuralink. Would you mind comment or so towards that?
What's so interesting about Elon Musk is he honestly doesn't care that much about the science, right? He just wants to do it and try to make it work, which is it's an approach that the academic community can be frustrated by, but it's also a valuable approach because if we just wait until we know everything, then we're waiting too long. And so his approach is to have thousands or millions of tiny electrodes that can be used to interface with the brain. Now, the safety issues haven't been resolved, the biocompatibility issues haven't been resolved. But that said, they're pressing forward with these studies, and I'm sure that technology, that nanotechnology that they're working with is going to be important. And there is a school of thought that if you make the electrodes small enough that the body won't mind them so much. So
is that true?
I don't think it's not proven, but it's possible.
Ah, interesting.
Yeah.
Okay. That actually tied to our last question in this section is what invention or applications do you envision will be developing in the future?
Right. So I do believe that in the we have good technology, or pretty good technology right now for helping people with paraplegia. I think that we will, in my lifetime, I hope, if I have a normal lifespan, I think that we'll begin to make progress on memory disorders like Alzheimer's disease, and that we will be able eventually to regrow damaged brain tissue, which is that's a holy grail problem for us, right? Because the central nervous system doesn't regenerate very well at all.
Exactly.
So if you have a stroke, you've lost function.
Yeah.
But there's no reason in principle that we can't regrow that brain tissue. It's just that we don't know how to tell the body to do
grow
it.
Okay.
Yeah.
Gotcha.
Okay, well, welcome to my lab. This lab is in a building that is devoted to interdisciplinary research in life sciences and engineering. It's called the Center for Interdisciplinary Research in Life Sciences and Engineering. And there are several of us. It's a sort of an open concept lab. The idea is to promote more collaboration, which has been very successful. So on this floor is myself. There's another BME professor, Shu Han, has her lab space here. Anna DeVore from our laboratory also has her lab space here. And we work together on interacting projects and have a great time, pushing the boundaries of neuroscience and neural engineering. But this is the room where we do much of our experimental work, and we have these home-built, complex home-built microscopes run by this big laser that's in the back here with these complicated laser paths and these curtained off areas. And we do a lot of our work, it's optical recording. And so we're basically exciting fluorescent molecules that we have often, that we've managed to introduce into brain cells. And those fluorescent molecules are reporting to us what the brain cells are doing, the level of activity, the electrical activity, which electrochemical activity in the brain. So this is the basis of our data set. We also use optogenetic techniques, which are techniques by which we can shine light into cells that have been transfected, meaning that they're expressing a foreign protein, and those foreign proteins make them optically excitable. So we can use optical methods, a microscope, to record activity and also to stimulate activity. So we use different colors of light to stimulate versus record. We can do them simultaneously. So this is a lot of the basis of what we do happens in this room. That room is used to make the electrodes, and it's mainly made for making electrodes. So we do a lot of electrophysiology, and we have to make our own electrodes. So everything has to be handmade. It's like being an artisan, And so that happens in there. And then we have benches, and you recognize all benches look almost the same, right?
Yeah,
so we have, we pipette, we make solutions, we weigh things, we stir things together. And let's see, yes, we do histology and things like that in this room.
Okay.
And we have another room in which we do recordings. I don't know, interesting, the data are exciting. I don't know that the equipment we use to collect the data is all that exciting, but you can decide for yourselves. Okay. Yeah, so we do these nuts and bolts recordings, biophysical nuts and bolts recordings in a room like this.
Yeah.
So we have these two setups, we call them rigs, and in these rigs we're recording biophysical details and doing biophysical manipulations. And Brandon is one of my experts at this kind of thing. He picked it up very, very quickly. And he's doing things like, we mentioned optogenetics. He's doing optogenetic in like little patches of brain tissue, like cultures. And so we take those patches of brain tissue and we try to understand the way neurons interact. At the very basic level, we do computational modeling to kind of build, that are basically our hypothesis about how things work. And we test the model until we manage to find out that it doesn't work, and then we adjust it. So we keep on cycling through that. So it's actually an important part of our work. And Brandon has a microscope here that allows him to see into the tissue so that he can lower those homemade electrodes I was talking about into the tissue. And he can do optogenetic stimulation and electrophysiological recordings. So what we're doing here in these experiments is we have the ability with this kind of microscope. We have electrodes that there's an electrode at the end of this. It's pulled glass with a salt solution inside it and a wire stuck in the back. And so we have a laser in the back that allows us to do stimulation, light-based stimulation. So we can basically drive these tissue cultures with light to open ion channels to provide this form of very flexible excitation to the network, and then record the activity in individual cells as we're doing that.
Gotcha.
Yeah, so we can do optical recording, electrical recording, and optical stimulation.
I feel like a stressor that students now face that was less bad for me was that the academic job market is challenging now. In my case, I just had to wait for the field to develop. I was a bit too early. So when I applied for faculty jobs, there were 2 biomedical engineering faculty jobs in the country open that year.
Only 2.
Boston University and the University of Utah.